So here we are on pages 72 and 73 to examine our eukaryotes. When we take a look at a eukaryote, like this one, which is an animal cell, what we're noticing is a distinct lack of a cell wall for this particular organism. You still have a membrane. If you're talking about a cell, there has to be a membrane. That membrane can be specialized. It can have these weird projections. It can be smooth. But the main thing that we really care about there is that that's where you have interaction with the exterior. So that membrane's got to be a good border that'll keep things out and keep things in. And it's going to have to allow transport of materials across itself in order for this cell to function properly. Inside the cell you can see the nucleus. There's a dark spot in the nucleus, which we call the nucleolus, and that's where ribosomes are starting to be produced. Those ribosomes don't necessarily have to stay in the nucleus. We can send them outside of the nucleus. Once the ribosomes are outside in the wild world of the cell, there's two locations they can be in. They can float freely in the cytoplasm or the cytosol, whatever you want to call it. And then some of them become associated with the exterior of this tube structure, which we call the endoplasmic reticulum. These tubes are actually made of lipids, and so what happens is you have a new compartment that's got an interior and an exterior. As we saw when we talked about a cell, to have an interior and exterior is significant because you can develop different concentrations of different things inside and outside, which means you can have specialized things happening inside that aren't necessarily happening outside. The rough endoplasmic reticulum, this tube structure that has all these little dots on it, this is primarily going to be where we are making proteins for export. These could be proteins that are destined for the extracellular matrix. These could be proteins like the lysozyme that you want to release into certain regions so that they can take care of invaders, right? Like those bacteria we just talked about. So anything we want to send outside of the cell, we're going to make inside this ER, this endoplasmic reticulum. Membrane-bound proteins, integral membrane proteins, things that have huge hydrophobic domains, we're going to want to produce here as well. And the reason is if you were to produce them in the cytoplasm, they wouldn't want to interact with anything they were nearby. Whereas if they are inside a membrane, they're going to be quite happy. Their hydrophobic region is interacting with other hydrophobic regions in that lipid bilayer. This is also going to be good for the cell because some of those proteins are going to serve as anchors to try and hold down some of our cytoskeleton. Some of those proteins are going to stick outside of the cell to receive messages and convey those messages to the interior of the cell without having to break through that membrane. Some of those proteins that sit inside the membrane will also serve as channels, like the aquaporins, which are channels for water, or like the sodium-potassium pump, which is going to allow for the transport of sodium and potassium across the membrane. Once we produce these things, produce these proteins, we have to send them for processing, and we're going to send them to an area we call the Golgi apparatus, which very frequently is described as a stack of pancakes, and as I told you in class, I don't really see it, but we do have a stack of membrane-bound compartments. And basically, you're going to have a vesicle drop off its cargo at this first compartment and pass it down the Golgi until it reaches the side towards the membrane, towards that outer cell membrane. So the vesicle goes from the rough ER to the Golgi, goes through the Golgi, and then from the Golgi it can exit the cell. We're going to call the area of the Golgi that's facing towards the nucleus the cis region, and we're going to call the area that's facing away from the nucleus the trans region. And the reason we specify these guys are different is because different modifications happen in each spot. Some modifications that we are expecting to see are things like the addition of sugars, our glycosylated proteins, right, the ones that have those sugars attached to them, are going to be very important in the extracellular matrix, meaning that they have to get outside the cell. We're also going to have some that will be stuck in a membrane, and those guys might also need some modifications to occur. And so that process we call maturation, and it's going to happen as it goes towards the trans part of the Golgi. Once it reaches here, and we've got our mature proteins, we're going to package them up again and send them off where they need to go. If those proteins are destined for outside the cell, that process we call exocytosis. Exo for outside, cyto for cell. So if they're destined for exocytosis, we'll send them from this region in the Golgi all the way to the membrane. And then at the membrane, the vesicle will fuse and release its cargo to the exterior of the cell. If the proteins that we've matured inside the Golgi are destined for some other job, for example, if they're enzymes that are needed in a lysosome to break down other substances, that vesicle will go and join that other compartment. A lysosome, as you know, is its own membrane-bound compartment. So the vesicle that's taking lipids from here, we're going to take lipids and proteins and package them up and send them off. And if they're digestive proteins, right, like proteases, DNases, lipases, we can send them to something like that, like a lysosome. That's exocytosis. In order for this cell to take nutrients in, sometimes it's going to take big chunks of nutrients in. I have a link for you guys down below that shows an amoeba eating. What happens there is that the amoeba surrounds its food and pulls it in. And then it takes vesicles full of digestive enzymes and throws it at its food so that it literally digests those cells inside it. We call that endocytosis, things going into the cell. And when you're taking a big bite, or if you're taking a little sip, it's going to be different. If you're taking a big bite of something from outside the cell, like a whole other organism, we're going to call that phagocytosis. If you're just taking a drink from outside the cell, we're going to call that penocytosis. So phagocytosis would be a larger substance. Penocytosis would tend to be just a little small liquid vesicle, liquid-filled vesicle that it pulls inside. Other organelles I wanted us to take a look at, we've got mitochondria. One of the key features of the mitochondria is that it's a double membrane system. And that's going to be important in just a minute. The features inside of the mitochondria, all the different names, I wouldn't worry too much about them just yet because we will be talking about respiration and spending a lot of time on respiration. And so the anatomy of a mitochondria is going to be more important then. What you can see next to the mitochondria in this diagram are these sort of weird pasta-looking structures that are at 90 degrees with one another. What these guys do is help organize the microtubules during cell division. This structure, which we call the centrosome, is not found in plants. They use some other means of organizing their cell division. They still use cytoskeleton, but they're going to organize it and have their organizing center be just a little bit different. When I said cytoskeleton, that's another organelle, not a membrane-bound organelle, but it's important nonetheless. It's going to help us keep the cell shape, right? So it's going to, for example, help the nucleus maintain its shape. It's going to help the cell itself maintain its shape. It's going to provide a highway for movement. Remember looking at the animation of the motor protein and talking about how the motor protein cost 1 ATP for it to take a step? It literally had to walk along something. In order for those motor proteins to work, the feet of those motor proteins need to chemically match up to whatever they're walking on. And so the cytoskeleton provides the road. And so kinesins, for example, what they do is they interact with microtubules. Dyneins also interact with microtubules, but the two of them will go in opposite directions. so that one can carry vesicles away from the nucleus, that's the kinesin, and one can carry vesicles towards the nucleus, that's the dynein. Other key features before we move on, we talked about lysosomes, we talked about endocytosis, penocytosis, phagocytosis, exocytosis, the nucleus, the nucleolus, the pores. We do have linear chromosomes here, and they're all scattered throughout that nucleus. We also have ribosomes, some floating in the cytoplasm, some on the rough ER. We also have a part of the endoplasmic reticulum that does not contain associated ribosomes. And so we're going to call that the smooth endoplasmic reticulum. And the smooth endoplasmic reticulum, it's going to take care of things like detoxification. So it would be important in places where you might be dealing with that, like the liver. It's going to deal with things like storage of calcium, generating lipids, right? Different cells and different organelles in the cells might expect to have different lipids.